Cardiac muscle contractions. Excitation-contraction coupling. These are the following steps in the excitation-contraction coupling in the cardiac muscle cells. The cardiac muscle cell contraction is initiated with calcium entering the cardiac muscle cell through voltage-gated calcium channels. This will cause the opening of voltage-gated calcium channels inside the sarcoplasmic reticulum, the SR, that leads to calcium being released from the sarcoplasm, and more than 80% of the calcium is stored inside the SR is then dumped into the sarcoplasm. The calcium is released into sarcoplasm from both the interstitial fluid and the SR. It then attaches to troponin and thus starts the cross-bridge formation between myosin and actin heads. The power stroke occurs with the myosin head swiveling and pulling on the actin, allowing the distance to shorten and therefore the shortening of the sarcomere. ATP binds to the myosin heads, release it from the actin. ATP is then split by the myosin ATPase, providing energy to reset for the myosin head. So remember CPRR. Cross-bridge, power stroke, release, reset. The voltage-gated calcium channels close in the SR and eventually allow no more calciums into sarcoplasm, and the calcium then is already in there must be removed via the CERCA, the calcium pump ATPase. They'll pump it back up, thus leading into the relaxation. So here are the following steps to make things easier. Step 1, during that action potential in phase 2 of the plateau, causes the calcium via the L-type calcium channels, also known as the dihydropyridine, DHP receptors, from the ECF into the ICF. Step number 2, this is the calcium-induced calcium release phenomenon. So as you increase the intracellular calcium concentration, this triggers release of more calcium from the storage of the SR, and the calcium releases from the ryanodine receptors. Those are the gates that are closed inside the sarcoplasmic reticulum. Steps 3 and 4 include calcium now binding onto troponin C that then frees up the whole actin and myosin to engage. Then that we have the cross-bridge cycling of CPRR. That follows the 5th step, which is going to be the amount of tension developed by the myocardial cells is directly relatable to how much intracellular calcium there is in the cell. Calcium enters via the plateau phase, leads to the calcium-induced calcium release. Calcium binds to troponin C, causes the cross-bridge cycling, and tension is generated. Relaxation occurs when the calcium is reaccumulated in the sarcoplasmic reticulum by the action of the sarcoendoplasmic reticulum calcium ATPase, CERCA. In addition, some of the calcium that has entered during the plateau phase of the action potential is now extruded out from the cell via the calcium ATPase and calcium-sodium exchangers on the sarcolemmal membrane. So you're able to get rid of the calcium inside the cell. As a result of this, there is going to be a longer duration of action potentials in the cardiac cell, and then the absolute refractory period is then prolonged. Effects of the autonomic nervous system on the heart rate. Factors that alter the heart rate are referred to as chronotropic agents. Some of these agents can increase the heart rate, and these are what we call positive chronotropic, and some of these can decrease the heart rate, what we call negative. Changes to the heart rate is directly due to the ability of acting upon the SA nodes and the AV nodes. The sympathetic nervous system releases norepinephrine and epinephrine that act directly upon the beta-1 adrenergic receptors that ultimately increase the calcium influx into the nodal cells. This is activated via the cyclic AMP secondary messenger system. Receptors utilize adenyl cyclase through the GS protein that will stimulate the increase of cyclic AMP, and therefore pKa. The SA node then produce an increase in the funny current, the I subscript F, and also an increase in the current of calcium, causing it to depolarize. A negative chronotropic decreases the heart rate. Acetylcholine released from the parasympathetic nerve fibers activate the muscarinic M2 receptors. M2 uses a subclass called G subscript K G proteins that inhibit adenyl cyclase that will then decrease a drop in the funny current to the heart cells. GKs also will then increase the conductance of potassium channels, therefore hyperpolarizing that diacetylic potential, making it harder and slower to fire another signal. With sympathetic stimulation, you can see that there are a lot more of these peaks. So there are 3 peaks in the same frame, whereas in normal you only have 2. It does this by making the pacemaker potential a little bit shorter. Whereas in the parasympathetic, it makes the pacemaker potential longer, thereby only generating 1 peak and 1 signal. and thus decreasing the heart rate. This is also why beta blocker drugs are negatively chronotropic agents. They block the beta-1 adrenergic receptors so that the epinephrine and norepinephrine cannot bind to them. Effects of ANS on the contractility of the heart. Contractility, also known as ionotropism, is the intrinsic ability for myocardial cells to develop a force at a given certain length. Agents that produce an increase in contractility are called positive ionotropic effects, and then agents that produce a decrease in contractility are called negative ionotropic effects. In the positive side, we have the catecholamines, epinephrine, norepinephrine. They will then activate two proteins via phosphorylation to increase contractility. The first one is phosphorylation of the sarcolemma calcium channels, thereby increasing the amount of calcium released from the SR. So as more calciums come in from the membrane, that will trigger the sarcoplasmic reticulum to release more. Number two is a phosphorylation of a protein known as phospholambin. This is a regulator of the SIRCUP protein. It regulates the sarcoplasmic reticulum calcium ATPase. When the phospholambin is phosphorylated, it causes a greater uptake and storage of calcium inside the sarcoplasmic reticulum. Negative ionotropic agents decrease cardiacally. The parasympathetic system and the acetylcholine have a negative impact on the atria. They activate the GI protein, which is a subclass called GK, which decreases contractility via two factors. The acetylcholine decreases the current of calcium and also the inward current of how much calcium is available during the plateau phase of the action potential. So less calcium means less signaling. It also increases the current of the potassium via the acetylcholine binding and shortening the duration of the action potential. For the sympathetic division, the heart rate gets increased by the beta-1 receptors. That leads to increasing in the funding current and increasing in calcium current. Parasympathetic will decrease the heart rate by acting on M2 receptors. It decreases the funding current, decreases calcium, but increases potassium current. The contractility over here, you can see sympathetic also increases the contractility by targeting the beta-1 receptors, by boosting up the calcium, so more calcium and phosphorylation of phospholambin. Whereas the parasympathetic will only decrease the atrias, targeting the M2 receptors, decreasing again the calcium influx, but increasing the current for potassium. Effects of the heart rate on contractility. Heart rate increases as contractility increases. When the heart rate decreases, the contractility decreases. The first phenomenon is called a positive staircase effect. This is also known as the Bowditch staircase, also referred to as a treppe. When the heart rate doubles, an increase in tension occurs because there's more action potentials per unit time. That means more calcium is entering in during the plateau phase, and there is going to be more accumulation of calcium inside the sarcoplasmic reticulum. That allows a more forceful contraction. Is that there's a staircase-like growth as you allow more calciums to go in, allowing more tension to be generated and more firing to occur. So there's a staircase manner of treppe that can be seen. The second phenomenon is called post-extrasystolic potential. This occurs when a signal is produced by the latent pacemakers, the non-SA node cells, the AV node, bundle of His Purkinje fibers, that can generate tension that is higher in the next beat contraction. What happens is that during the extrasystolic contraction, there is a slight drop in tension because there's not enough calcium, and most of the calcium gets stored back up, building to a next contraction that is higher. So the post-extrasystolic contraction is greater than the extrasystolic contraction that occurred. And this is a phenomenon that occurs when the latent pacemaker signals through. Effects of cardiac glycosides on contractility. Cardiac glycosides are a class of drugs from the foxglove plant, also known as Digitalis purpurea, and they act as a positive ionotropic agent. They increase the contractility of the heart. The major therapeutic uses of cardiac glycosides is in the treatment of congestive heart failure. This is a condition that's characterized by there's a drop in contractility of the ventricular muscles, a negative ionotropism. When heart failures occurs, there is not enough tension to be developed, and therefore it cannot eject the same normal amount of stroke volume of blood into the aorta. And this is very serious and of course life-threatening. So the mechanism of cardiac glycosides is as follows. They inhibit the NAK ATPase. The NAK ATPase is located in the cell membrane of the myocardial cells. Their job is to maintain the potassium and sodium balance. What the cardiac glycosides do is they will inhibit the extracellular side for the binding of potassium. thereby inhibiting the NAK ATPase, leading to a higher amount and increase of intracellular sodium concentration. Because there's more sodium concentration now inside the cell, it messes up calcium-sodium exchanger, and therefore less calcium is then pumped out of the cell, which causes a buildup of calcium inside the cell. So over here, you can see that once you block off the potassium, channels are not working anymore. As a result, sodium increases in the cell. This will then cause you to have a disruption to the normal Na calcium ATPase. Because there's so much sodium inside the cell, the calcium-sodium ATPase is not going to work anymore. As a result, calcium builds up in the cell, leading to a positive ionotropic effect.